Well, hello everyone, and a very warm welcome to the first in our new series of Genome Lates, brought to you by Wellcome Genome Campus Connecting Science. I'm Ken Skeldon from the Campus Public Engagement team, and over the next few months we'll be discussing and exploring big themes in genomics and biodata, inspired by this year being the 20th anniversary of the first draft of the Human Genome being published. All our speakers have connections to the Wellcome Genome Campus, which hosts the Wellcome Sanger Institute and the European Bioinformatics Institute. And indeed, it's a double anniversary, as 30 years this autumn marks the start of the International Human Genome Project effort, which this campus was played such a leading role in. We've set up the series very much around conversations because we want to explore the science while also hearing the personal reflections from our guests around genomics and biodata, including where new discoveries or technologies might take us. And importantly, we want to hear from you, the audience. So we'll be leaving good time for questions and discussion. If you do have a question or a comment that you want to make, then please use the Q&A feature on the Zoom window. Now, you'll either see that at the top or the bottom of the screen, depending on the device that you're using. Don't worry if it looks like you're the only one asking a question or that box is empty, because what will happen is your questions will come into a feed to us and to the speakers. And my colleague Beth, who's working behind the scenes, will try and cluster those so that we can get through as many questions as we possibly can. There's over 100 people of you here so far, more joining, and so we hope to have a fantastic event. So now just to introduce this evening's speakers. So we're delighted to launch the season with Professor Sir Mike Stratton, the Director of the Wellcome Sanger Institute and CEO of the Wellcome Genome Campus. Mike's primary research in cancer included the identification and mapping of the BRCA2 gene, which plays a major role in breast cancer susceptibility. In 2000, Mike initiated the Cancer Genome Project at the Wellcome Sanger Institute, which carries out genome-wide searches for DNA changes that could play a role in human cancer. Through these studies, he discovered DNA mutations in the BRAF gene in malignant melanoma and several other mutated cancer genes in lung, renal, breast, and other cancers. His work continues to reveal the basic patterns of change happening in the DNA of cancer genomes and how mutational and repair processes work. Mike's a fellow of the Royal Society, and he was awarded a knighthood in 2013, recognizing his long-standing contributions to genomics. This evening, Mike is in conversation with Kat Arney, and Kat is an award-winning science writer, broadcaster, and public speaker. Our new book is called Rebel Cell, Cancer, Evolution, and the Science of Life, and is latest in a series of popular books on genomics, which include How to Code a Human and the critically acclaimed Herding Hemingway's Cats, Understanding How Our Genes Work. Kat also presents the popular Genetics Unzipped podcast for the Genetics Society and has fronted several BBC Radio 4 science documentaries, including the recent series Ingenious, looking at the stories behind our genes. So without further delay, I'm delighted now to invite Kat and Mike to the Zoom stage to explore reading the Book of Life, what has genome sequencing ever done for us?
Great, thank you very much, Ken. And hello, Mike, it is really wonderful to chat to you.
Hi, Kat, it's wonderful to be here.
So let's start by looking at, I'd really like to just get a sense of the scale of where we've come from, because we went from the Human Genome, which was, it was about 5 or so people all contributed to that genome, to where we are today. And I remember I was doing my PhD around about the year 2000, and we were inching our way across the genome in 200 base pair little chunks. So can you give me an idea of from what it's looked like from your angle of the scale and the pace of change since those days?
Well, thank you, Kat. As you say, in 2000, the draft human genome sequence was released to the world. And that had taken, many organizations, huge effort. The Sanger Institute did about 1/3 of it. and there were hundreds of people working together with robots and computers to do that. But it was an international effort to do that one reference genome. Where we are today is that we're certainly on our way to sequencing hundreds of thousands, the low millions of whole genomes. And that has happened because of really quite extraordinary changes in technology that have occurred in the meantime. And if you look back at that genome that was sequenced, the reference genome in 2000, maybe that cost, I don't know, 100 million to $500 million. The cost of a genome nowadays is about $500. So the cost of producing a whole genome sequence has reduced by 100,000 to 1 million-fold through those changes in technology.
So we talked about the original, the Human Genome Project, but given now how many genomes we have, can we really talk about the human genome? Now that we've got hundreds of thousands, what are we starting to discover about what this human genome supposedly looks like?
Yeah. Well, the human genome, it's about 3,000 million letters of DNA code, and each of us have 2 copies of that, one from our mother and one from our father. And yes, it's different between 2 people. It's about 1 in 1,000 letters of code differs between 2 individuals. So that may sound like that's a rather small proportion of the genome that differs between us. But of course, that amounts to about 3 million differences between each of us. And those 3 million differences in sequence are what account for the differences in the way we look, possibly to some extent in the differences we behave, and our predispositions to various different types of disease. And really the last 20 years, one of the major outputs of all that sequencing of our genomes has been to catalog, in the first instance, those differences between us, those sequence differences. Some of them are quite common. So half of the people in our room now may have one variant sequence and the other half may have the alternative. And some of those differences are very, very rare. So it may only be, you know, 1 person out of 1 million that would have that particular difference in gene sequence. So we've cataloged them. And now we're deep in the business of working out what those differences do.
Exactly. It's not what you've got, it's what you do with it. And I've always been really fascinated by I like to think of it as the black box between you've got the code going in, the DNA, just the letters, the instructions, and then you have life coming out the other side of it in all its glory. So how are we starting to unpack how you go from this code to, you know, what does it take to build a human, and then what happens when that doesn't work out, when that does go wrong in disease or different conditions?
Well, there are many ways of going about that, but the one that I think has been used in various different contexts is to basically sequence people with known phenotypes. So we've got our reference human genome sequence, and then we sequence a number of different people with a variety of different diseases, a variety of different normal traits, and we essentially do correlations. We say, when we look at people with this particular disease, we find that there are more mutations in this particular gene than there would be in the normal population. And by doing that in disease after disease, common diseases such as diabetes or Alzheimer's, rare diseases that maybe only occur 3 or 4 times in the world each year, we're building up that catalogue of knowledge of how differences in the human genome, sequence differences, contribute really to the wide range of human disease and indeed to the range of human normality too.
That's the thing, you know, I like to think everyone is a mutant in their own way. We all have so many variations between us. And it does seem like a really challenging task to compute that information. You have so much diversity, so many things you could measure about a human, about the phenotype, the characteristics. I mean, how are we starting to from a computational point of view, integrate all this information?
Well, you know, there are significant computational problems with it in just handling these amounts of data and processing them. Most of, you know, for me, looking back 20 years, the notion of having hundreds of thousands of whole human genomes was an unimaginable thing to think about. And actually, I remember very clearly just when we switched to the new technologies here at Sanger in about 2008, I got a deputation from the IT people saying, you can do this, you can generate all that data, but we can't guarantee you that we're going to be able to handle it. So from that very simple perspective of simply managing it, it has been a real roller coaster right through.
Yeah, any of us who remember doing a blast search overnight, you had to, you know, do these things in hours of time.
Yeah, absolutely. Then integrating it with all the various different types of data that give us information, give us insight into what the bits of the genome do. Those are further kind of integrative computational challenges. But the basic principles are pretty simple. What we're looking for is an excess of mutations in a particular gene, let's say, associating those with a particular phenotype. And that's a very simple precept on which to build, even if it becomes complex in its implementation.
Here's a little sneaky question. What's the difference in your mind between a mutation and a variation?
There is no real difference. Those words can be used in some context differently. So a variant is a rather neutral word, which just means a sequence difference. A mutation can mean that as well. But in some contexts, speaking of a mutation tends to get more associated with a disease causing change. But ultimately, one can use them interchangeably.
I remember speaking to Anna Middleton, who does a lot of the genomics and ethics work, and she said that some people quite like the idea of being a mutant. It sounds like being one of the X-Men or something like that.
Well, and you know, it's absolutely right what you said earlier, that we are all mutants. In fact, going back to the question you asked right at the beginning, is there a single human genome? There is no single human genome. And if you do the sums, actually, it is highly likely that every single one of those 3,000 million positions of in the human genome is mutant, is variant somewhere amongst the 7 billion people on the planet. So there is no absolute, for no position in the genome is there just one base that affects, one letter that is with us all.
That's amazing to think about that. And kind of zooming in a bit, we've talked about the huge variation in humans that we're starting to see, and through the ability to sequence more and more genomes from people, and you do that by taking a saliva sample or a blood sample. But now the technology, and I guess this was absolutely unthinkable 20 years ago, to be able to take a single cell and look, now we can look at the genome in it, but also to look at things like how those genes are being used, how they're being read, look at single cell transcriptomics, what this cell is up to. So now that we can do that, what are we starting to understand about how this one set of instructions, the genome, is actually being used in all the different sorts of cells in our body?
Well, the first thing that approach is enlightening us is a very fundamental question, which is how many different types of cell are there in the human body? So our knowledge base for this really comes from microscopy, data that was generated 150, 200 years ago, where people looked down the microscope at human tissues, and they could look at a muscle under the microscope, and they would see a particular type of cell, and they'd call that a muscle cell. And they'd look at the brain, and they would see certain types of cells that looked rather distinctive, and those were nerve cells. And they could look at fatty tissue and see fat cells, adipose cells. And so you build up this set of cells. And indeed, this catalogue of cell types is based on microscopy for the most part. But of course, the reason why those cells look different is because each of those cell types is using a different subset of the 20,000 genes in the human genome. So a fat cell may be expressing 10,000 of those. A muscle cell may also be expressing 10,000, but a different, or an overlapping, but not identical 10,000. And it's those differences that are responsible for the different cell types. And because that is the fundament of why cell types are different, that now we can take individual cells, single cells, and we can sequence their RNA. That's what you called single cell transcriptomics. And we can work out directly which of the 20,000 genes in the human genome is being expressed, in other words, is being used in that single cell, and which ones are not. And then we go to the next single cell and do the same thing. And so now we build up these huge data sets from single cells, and we can say, well, this cell has got these 10,000 genes on, and this cell has got pretty much the same set, so probably they are the same cell type, but this other cell that we found in this sometimes randomly harvested set of cells from a tissue looks rather different, so that must be a different cell type. So we're in the business of re-identifying, recataloguing the cell types in the human body. And indeed, that it goes for normality, and we're beginning to see many more cell types than were available to us by looking down the microscope. We're seeing great subtlety in the way that, in the differences between certain cell types. We're beginning to see that in disease, there are additional cell types that don't quite match those that were seen in normal tissues. And then over the next 10 to 20 years, we will take all that information and not just have a catalogue, a new classification of cell types, but we'll be able to look at those, which genes are expressed in the tissues. Now, again, coming back to down the microscope. And we'll be able to see really fundamental patterns of biology, that this cell expressing that 10,000 genes tends to be more close to that other cell, which is expressing those other 10,000 genes. So we will build up really refined understanding of how human tissues and organs are put together.
I wanted to just ask you about some of the technical advances that have made this kind of thing possible, because I've been to see some of the robotics at the Sanger that enable the single cell work to happen, and it's incredible, like being able to measure tiny, tiny amounts of fluid, being able to have robotics that ensure that everything happens the same. In terms of things like the sequencing and also in the sample handling as well, we talked briefly about the computational side of things, but like technically, what do you think has been really important to enable this kind of single cell resolution stuff to happen?
There have been, there's been the ability to isolate single cells, the ability to use the RNA from a single cell, which of course is a very small amount of it, to process it without it degrading. RNA is very prone to degrade, DNA is very robust, RNA, and to basically process it in such a way that by sequencing it, we can retain those levels of the ability to detect those different levels of expression from different genes. It's been a whole series of technical advances, but it's all down to the ability to do very large amounts of, multi-parallel sequencing, technologies that were developed really between 2000 and 2008, which allow us to, which did reduce the cost of sequencing by this extraordinary 100,000 to 1 million fold.
And so that's kind of the next generation sequencing. So what do you think's the next generation? You mentioned being able to look at cells in the context of tissue. Do you think we'll be able to sort of do that soon?
Yeah, I mean, I think those technologies are coming along. There have been some quite remarkable advances in those. It's how far we'll be able to go. We can already see differences if we look at just, 5, 10, 100 genes. Whether we will be able to get to the position where we can look down the microscope and say, this cell has got these different levels of 20,000 genes, and the next cell has got a different set of levels of our 20,000 genes. That's quite a long way away. But yes, that sort of in situ sequencing is certainly developing, and it is, if you like, one of the major next technological boundaries to be overcome.
Well, when we come back in 20 years from now, I think, given the rate, perhaps we can talk about that. But I wanted to talk really, because being able to look at the number of human genomes, basically being able to do genomics at that scale, being able to look at tissues down to this fine level, it gets to this question of what is normal. And I really, really want to talk about this next bit, because it's really relevant to the stuff I've talked about in my new book, Rebel Cell, which is about what is a normal cell anyway? And the discovery that really blew my mind, and it was work that came out of the teams at the Sanger, was showing that even supposedly normal cells in our body that you would think are not mutant in any kind of way, that normal healthy cells are peppered with what we would call mutations, and in some cases what we would call cancerous mutations. So what is normal, and what is this genomic sequencing now starting to tell us about the state of our tissues as we progress through life?
Well, we certainly are having to question the use of the word normal, and to use it carefully. The fact that cells accumulate mutations throughout life is, I think, it has been shown recently, but it's probably not so much of a surprise. I think most people did assume that as cells divide, the DNA replication mechanisms were not going to be perfect, and that the occasional mistake would be made, and a mistake is a mutation. So that as cells continue to divide through life, that they would acquire mutations. It actually has been much harder to study normal cells than it was to study cancers, for technical reasons. But those technical obstacles have been overcome, and that it's been possible over the last year or two to do that. And indeed, we and others have found that over life, cells accumulate mutations, more or less randomly distributed through the genome. They don't do it all at the same rate. Some cells acquire mutations pretty slowly during life. Other cells acquire them much more quickly, and we don't really completely understand why those differences exist. But yes, different cell types do it at different rates. What has been a little more of a surprise is what you alluded to, as that some of the mutations that we're finding in normal cells are actually the sorts of mutations that we might expect to find in cancer cells. So we now know through sequencing of tens of thousands of cancer genomes over the last 20 years, that there's a set of about 800 genes, which if you get the right mutation in that gene, in the right cell type, it will contribute to converting that cell into a cancer cell. And we call those cancer genes. And we believe that all cancers are caused by mutations in those genes. What was surprising was to find that actually we do find those mutations present in cancer genes in normal tissues. And indeed, in quite a lot of cells in normal tissues. In the early studies, we found in the skin that maybe in a 70-year-old man in their eyelid, heavily exposed to ultraviolet light, so very mutagenized, that maybe 50% of the cells had 50% of the cells had mutations in these cancer genes. And these cells were growing out as little clones. basically, these are little benign tumors, but 50% of the apparently normal eyelid was composed of these thousands, hundreds of thousands of microscopical benign tumors that you can't see down the microscope. So that was the skin, you know, lots of ultraviolet light. The next study looked at the esophagus. And there the expectation was that perhaps there was going to be less of this going on because you don't have ultraviolet light causing loads and loads of mutations in the esophagus. And indeed, the overall number of mutations in the esophagus was lower, but the number of these clones with cancer mutations in them was the same. So again, as you get older, you get to the stage that 50% of your esophagus is made up of these multitude of little clones that are early cancers. And then the final one, if I can just say, was the endometrium, which is the lining of the womb. And in women age 60, actually it turns out that 100% of the cells already have one of these cancer driver mutations. In fact, some of them have 2 or 3 and are already down the line towards becoming neoplasms, becoming cancers. So the whole of the endometrium is basically made up of these thousands of benign tumors. But you look down the microscope, it looks perfectly normal. And of course it is normal because everybody has it. That is normality.
I found this fascinating when I discovered this. I went to see Phil Jones, who did a lot of the skin work, and he sort of peered in my face and said, "Your skin is a patchwork of mutation."
Yeah.
It was amazing. And I'm always curious now about where do we draw the line between what is sort of normal, for want of a better word, and what is cancer, sort of not just what is cancer, but when is cancer? And this idea that actually it's an evolutionary process from picking up certain changes, certain mutations, but there's got to be something else that is enabling these sad cells, these damaged cells with changes to actually go the full way and become a cancer. And I'm curious, you mentioned the endometrium because there's the condition endometriosis, which is where you have tissue in the body that's, you know, it has these changes in it, but we would not call it cancer, but it's a tissue that's gone around all over the place and causes a lot of problems, but that's not cancer. So it kind of like, what is cancer and when is cancer? I think this kind of work really challenges our understanding.
Yes. I mean, we defined cancer on the basis of the way it behaves. And we know that now that cancers were always rare events. If someone gets a cancer, they usually get 1, unusually 2 or more. What we're finding is that actually most people have hundreds of thousands, if not millions, of these microscopical benign tumors. So basically, it is a very rare event for one of these benign tumors to convert into a malignant tumor. And of course, like many things in science, when you think about it, actually you knew about that before, because we have a window on this on our skin, and all of us have nevi, have moles, little pigmented spots on our skin, and those are small benign tumors. Most of us do not get malignant melanoma, but when malignant melanoma occurs, it does come from one of those moles. So you've got this extraordinary hierarchy of hundreds of thousands, millions of benign tumors that we acquire, which don't cause us any trouble at all, really. And in fact, for the most part, we're completely unaware of them, even if we look down the microscope at the tissue. And then very occasionally, one of those clones makes that final step to start behaving badly in the way that a cancer does. We can begin to see what it is that is doing that, which is that actually on top of these driver mutations, what you often need to get is a degree of disorder of the genome of a cell, which then allows the cell to go through the gateway to become a cancer cell. And that disorder probably means that the cell is generating many, many more of those driver mutations. So it's that genomic instability of one form or another that then allows the cell to go that final step to become a cancer, fully fledged cancer cell.
They're basically just like its genome loses it. Like it just loses
exactly that. And they do look absolutely wild. They really are completely disordered and can, in many cases, continue to get more and more disordered.
I could talk about this all day, but we do want to move on a little bit, and maybe we can circle back to the cancer thing. But one of the other things that the Sanger is now up to is, you know, why stop at humans? Why stop at tissues? Let's sequence all the things. So as well as focusing on ourselves from the Human Genome Project, the Sanger is also looking at other species. So tell me a bit about the drive to look at the genomes of other species. Which ones have been focused on? Which ones are going to be the focus in the future? And are there some that are super interesting but overlooked?
Huh. So probably from the first moment that Fred Sanger invented his method of sequencing and was able to see DNA sequences, you know, his mind and that of others around him will have raced and will have thought, well, one day we might be able to do that human genome, and perhaps one day we may be able to do the genomes of all the species on Earth. That's what they might have thought. Well, it took 20-something years to go from having that, you know, fanciful insight about the human genome for us to get the human genome after they invented their method of sequencing. It's taken a little bit longer for us to start thinking seriously about sequencing the genomes of all of life on Earth. And like so many things in this regard, it requires a technical advance to make you start considering it seriously. And the technical advance here has been the ability to get DNA sequences that are not fragments of 200 or 300 letters of code, which you then have to piece together, but they're much, much longer. Thousands of letters of code, even tens of thousands to hundreds of thousands, reads of that sort of length. Because when you get reads of that sort of length, then you can begin to overlap them really quite easily and automatically assemble a genome. So you basically take your DNA, you put it into these new sequencing technologies, you get sequencing reads that are tens of thousands of bases long, you put those into a computer, the computer works out which of those overlap, and on a good day, what it spits out at the end is the fully assembled genome of that species. At the moment, we did not so many good days of that nature, but one is getting there, getting close enough to think, well, actually, this is something that we should now be investing in, because only by doing it, only by pushing the boundaries and really wanting it to happen, will we make it happen. Will we improve the sequencing technologies, and we'll improve the computational technologies to allow that assembly of the whole genome to be done better. So,
I was going to say as well, because that's imagining that you're taking a sample from, say, one animal or plant, but now you could do things like scoop up a cup of seawater and throw that through the sequencer and see all the different bacterial species. So I guess that is, again, the function of being able to read long bits and deconvolute all the different species that are in just kind of quite a mess of a sample.
Well, that's right. So that's called metagenomics. And basically it makes one imagine that one could be sequencing ecosystems and measuring all the components of an ecosystem and their contribution to that ecosystem through that sequencing, and then looking at how that ecosystem changes under different circumstances, and particularly under the circumstances that we've created in climate change. But, you know, coming back to the genomes of the species, we're not even going to think about too much at this moment, the bacteria and the viruses. There are probably more of those than we can imagine. And indeed, it's not quite clear what a species is in that regard. But of so-called eukaryotes, where there is a nuclear membrane, there are somewhere between, I think, 1 million and 2 million where there's a species name all around the Earth. And so the Earth Biogenome Project has said, let's start thinking about sequencing those 1 to 2 million species that we know about on the planet. Probably, I mean, almost certainly that's a vast underestimate. There are probably 10 million or more than 10 million species to be sequenced. So where do you start, right? That's a big task. Collection of all of those, I mean, that hasn't been done. As I'm implying, we don't know the names or even the existence of most of the species on Earth. So where do we start? Well, one quite good place to start actually is the UK or the British Isles, for a number of different reasons. One of those reasons is that actually the cataloguing of the species in the UK and in British Isles is actually pretty good compared to many other places in the world. Another reason is that we are pretty temperate, so we don't actually have so many species. If you don't have the biodiversity, if you go to Madagascar, there is a lot more biodiversity, a lot more species than we have. So it's a more digestible morsel in the UK. So we know quite a lot. We don't have so many of them. We have a great naturalist and scientific community that knows where to find them and to collect them. And we also have, you know, a Genome Institute that's willing to give it a go, and the Wellcome Trust that's willing to sort of support getting underway. So there are 66,000 known species of animal, plant, and fungus in the UK. And we have started to make that collection with the wider biological community of naturalists and lay people that are interested in collecting, and to make those collections and to start sequencing them. And we would like to get that project done in 10 to 15 years.
Wow. And I do have to ask, like, it's very cool and it's really exciting, and it's kind of quite a nice piece of scientific stamp collection. So like, let's collect the genomes of everything. But there's sort of the broader, like, why? What will you do with them when you've got them?
Yeah. Well, I think the first part, that it's cool, is pretty good. It is pretty cool. It's a fundament of the knowledge of the world that we live on, what species are there. And I would say that that's a pretty good goal in itself. I think we can then use them for two things. The first is about climate change. It is about monitoring the environment, the living environment, through understanding the organisms that are there and using their genome sequences in a way that you're alluding to sort of see what is happening to our world as we push it to the extreme, which is obviously what human beings are doing at the moment. So that is to do with conservation. That is the first aim of having all our species. The second is a longer term one, which is really about knowing nature's toolbox. We will there have, by sequencing the genomes of all species, we will have the end results of all the experiments that nature has done in exploring all the ecological niches on Earth. And we will have nature's full toolbox. We are entering into an era, slowly, of thinking about, you know, the creation of new forms of life. So knowing about nature's toolbox, knowing how nature has dealt with all of the ecological niches it has explored, is the fundament to thinking of that even greater sort of initiative, exploration of synthetic biology that will come in the future.
I was going to move on to that. I just want to remind the audience that please, if you have questions for Mike, we'll be coming to questions soon. So do pop them in the question and answer, and we'll come to that soon. But yeah, Mike, you mentioned the concept of synthetic life, and that's almost where I want to take this to, is that we've had for 40 so years the ability to read genomes. And I guess if you count the invention of restriction enzymes, the kind of molecular scissors in the 50s and 60s, we've been able to kind of cut and stick bits of DNA together. But where are we now with our ability to edit and even create DNA? Where are we getting to, moving from reading to editing and writing the genome?
Well, there have been technologies for, a decade or two now that enable us to mutate genomes and to mutate them in cells, so the cells have a changed genome. The advent of the CRISPR technologies in the last sort of decade or so has meant that that is much, much easier to do, can be done much more targeted, directed fashion, much more refined. And so we have, possibly not at will, but almost at will, the ability to change the genome more or less in any place that we want to in a living cell.
On a good day.
On a good day, yeah, absolutely. That's quite correct. So that's an extraordinary achievement, and I think will have impact in many areas of, for example, what used to be called gene therapy, in modulating, changing the genome in people who have genetic disease, particularly those that have somatic genetic disorders. But alongside that, there have been major changes in the ability to actually synthesize DNA, and really quite long stretches of DNA. So thousands to tens of thousands, even hundreds of thousands of letters of code being synthesized. And there you can go beyond changing one or two bases, causing a structure that was there to rearrange itself a bit. You can really consider reorganizing the genome in first instance in a way that you can absolutely direct. And then, yes, you can think of the next step of just creating completely new genetic sequences and and what that might be able to deliver and where you might take that.
So that would be expanding the genetic alphabet, I suppose, going from the A, C, T, and G that we have and the sort of various chemical modifications we know about those four bases to things that, are still bases and would fit within the double helix, but are different. They're different chemicals.
So you can do that as well, but what I was really referring to was, in the first instance, was to, just make new DNA sequences that have never been seen in the whole of nature.
New genes, completely new genes.
Yeah, in principle, a mind experiment. But then, yes, you can certainly create what you've been talking about, which is the idea of new bases and new ways of using DNA to synthesize new types of, macromolecule. Proteins are the ones that are synthesized from DNA, from RNA, but actually one could think of, using that whole machinery to produce completely new, different types of macromolecule using biological systems.
Yeah,
so,
this
amazing
whole field
sort
of synthetic biology.
Yeah,
the whole field of synthetic biology. I think what most intrigues you or excites you, if you could build a gene to do something, anything, what would you make a gene that could do?
I'm gonna be
useful.
I'm gonna do the trick of the stumped interviewer to ask you that question, Kat.
Oh, what would I like? I would definitely like to have a gene that turned, I think, at the moment, discarded masks and gloves around my town into something benign.
I see.
Getting rid of the worst human waste that's polluting the planet, I think, would be really useful. So we're about to go to Q&A, but I will just pick up on what you said earlier about the Wellcome being willing to fund the kind of research that's gone on at the Sanger and obviously the Sanger Institute itself. Now, how lucky are we here to benefit from the Wellcome's, I guess, vision in supporting this program from the start?
Yeah, no, we are extremely lucky. We are extremely lucky in many ways. Yes, you can just give the numbers of what Wellcome has invested into, well, just this the Sanger Institute and Wellcome Genome Campus. That's something like 3.5 billion and growing over the 25 years. That has given us the ability to, not just generate the genome sequences, but that's enough, but also to have the influence as to how they're used. It was Wellcome, which took the position that the human genome should be open and for everybody to use. And it was Wellcome's independence that allowed it to say, well, we're going to sequence the human genome, whatever anybody else does, and we're going to make it open for everyone to use. So to have that sort of influential, independent, bold thinking in a funder, that's a really, a privilege to have as a recipient of funding, but we're privileged in the UK, and indeed we're privileged in the world.
Yeah, it's such a fantastic place. I did my undergraduate projects at the Sanger Institute campus. I was working in what was then the Human Genome Mapping Project, and it was very weird to come back a few years ago and see how much had changed and how much it's grown, and just the banks of sequences. It's a phenomenal place.
Well, thank you very much for that. And we are actually looking to expand the campus to 2 or 3 times its size over the next 25 years under the headline of genomes and biodata. We believe.
Well, you need to build a pub there, so that's.
And we're intending to do that, actually. We're intending to have housing and amenities. A pub hasn't been specified, but no doubt will be considered.
So we're going to move to a few questions now. So we've got a question here from Adrita, who's a secondary school student in East London, who asks, what information is coded into DNA? And I think we talked a bit about the kinds of things that DNA and genes do. It gives cells their identity. So maybe thinking about this idea of information, what sort of information about how to make a human is in our DNA, and maybe what sort of information isn't in our DNA as well? If you've just got the DNA sequence, can you infer everything about an individual? What can we tell and what can't we tell?
Well, the DNA sequence does have the basic instructions for constructing, for making a human being. It has the 20,000 genes, the proteins, which make the proteins, which are the components of the molecular machine. And then it has, alongside those, the regulatory elements, which tell a cell, well, you should be making this, expressing this gene and not that one, and that full set of instructions. It's like having the software that you put into a computer that makes your computer function as a word processing machine. It won't do it unless it's got that software, and that software is the genome sequenced with all the instructions in it. But we do all experience the environment as we develop and as we grow, and it's pretty clear that being in the environment does change us. It does influence the way that we behave, that we think, and it can also influence our bodies and our metabolism in terms of our lifestyle and what we expose ourselves to. So the output, the final human being at age 70, at the end of life, you can't predict everything about that individual from the genome sequence that was constituted by the sperm that fertilized the egg 70 years previously.
This sort of ties into another question we've got from someone called Woods. And every time I give a public talk about genetics, I play kind of question bingo. I can always guarantee which questions will come up. And this is the epigenetics question, ding, which is how much is known about epigenetics, how genes are switched on or off? How does this change prior, during, and after a disease? And this is kind of a bit about what you just talked about, the influence of the environment on which genes are switched on or off or where, and how we respond to the world around us, I guess.
Yeah, so there is an increasing amount known about the modifications, if you like, of DNA that give it a structure and that do determine which parts of it are active and which parts of it are inactive. That's been a huge kind of revolution in understanding all of that over the last 10 to 20 years, really. It is known that elements of that can be influenced by external exposures and lifestyle. So, for example, smoking can change the modification, methylation of cytosines in DNA, which is a key epigenomic change. It doesn't change the actual letter of code necessarily, but it just changes the modification. And so there is a way through epigenetics and through the epigenome and through modification of DNA to change the way in which DNA is expressed. This is not my area of expertise, and I don't know the extent to which we have pushed that. But I think it has been controversial. The really controversial thing has been, can you epigenomically modify the genome and have that transferred into the next generation? And there have been quite a lot of toing and froing on that, and I don't think there is a consensus that actually is happening, but it has been a controversial area.
I would point anyone who's interested in that. We've just done an episode of Genetics Unzipped, the podcast about epigenetics, and it kind of looks into some of the experiments here and some very cool mice with little white gloves on. So I would just quickly flag that. Genetics Unzipped, go and listen to that. We have an interesting question here, which is, how do you know you have the full genome of another species if there's no reference standard? And I think sort of an adjunct question to this is like, how do you know what's a species anyway if there's so much variation? Like, how do you decide that's a species going on its genome, and how do you know that you've got the whole genome of a new species if you've never sequenced it before?
So I think, well, I don't think you will always know, actually. I think that will be potentially a problem, that you can't always know that you've got the whole genome. There are some elements of genomes that we will be able to identify through other means. For example, it is possible, of course, to see how many chromosomes a particular organism has. And if the organism has 11 chromosomes and we are able to assemble the genome of that organism into 11 pieces, that would suggest we've got all the chromosomes. It doesn't mean we've got all the genome, but let's say we can go from one end to the other of each of those 11 pieces. We may well be there, but we will never be absolutely sure. Yes, if we were to sequence 1,000 of those species, we would get a better view of what bits we may be missing out in any individual. So I think there will be uncertainty, but we will have, how can one put it, for most organisms, a good approximation, and we will build a set of tick boxes that we can use to say, well, probably this is nearly complete.
There's a fantastic question here from another attendee, anonymous attendee. It says, sociologist Reardon in the, I'll come back here. Sociologist Reardon in the Post-Genomic Condition poses the question about who are the people that will embody the benefits of genomic innovation. How is the Sanger attending to the issue of fairness so that underrepresented and underserved populations also benefit from the advances? And I guess this ties into the issue of genetic diversity. I've done a lot of work looking at that most of the information in our genomic databases is from people with European ancestries, and there's an awful lot of people in the world who do not have European ancestry. So what sort of areas are going on here?
Yeah, no, that's absolutely correct. And there are substantial initiatives in Africa that are being coordinated within Africa to sequence genomes from across the continent. Obviously, those, it is a question both of diversity, but also in that, going back right into the start of human history. So it's a scientifically really important thing, and it's with respect to health and bringing the benefits of genomes. Absolutely, we need to see sequencing of as many populations as possible to the level at which we can extract the sort of information that we to use for population benefit. It's a really fair question. and it's, as you say, most genomes are Caucasian, but there is work on the way to rectify that.
There's a lovely question here from Roshan De Silva. You talked about the ability potentially to engineer whole chromosomes, to write DNA, write new DNA. And they ask, could we add chromosomes using genetic engineering for those who have too many or too few chromosomes, like, for example, in Down syndrome? So is that the sort of, what could you do if you could build a chromosome? What could you do with it?
Well, in the first instance, well, there are a number of things one could do. I think in Down syndrome, where every cell of the body has got the trisomy, it is difficult to see how one would use synthetic genomics to rectify that. But I think in first instance, this will be a tool for us to experiment with genome structure, to understand how, if we reshuffle things in the chromosome, what difference does that make to how the chromosome will work. But there could be situations in which we do reconstruct parts of chromosomes to get past deletions, for example, that have caused certain types of disease. And that could be the case, for example, in certain tissue-specific diseases, such as thalassemia or diseases of that nature. It is still science fiction, but those are the opportunities that could be offered.
Yeah, I get very excited about the ideas presented by genetic engineering. Then you remember you've still got to get the stuff into every cell you need to get it into. Yeah. So, you can do it for blood disorders reasonably easily.
Well, that's right.
How do you do every muscle if you're looking at something like muscular dystrophy?
Well, people are getting it into every cell almost certainly is going to be a non-starter. It's all about getting it into a significant subset of cells. As you say, with blood cells, you can take it, a sample out of the body, you can engineer it out of the body and put it back in, and you can help it take over, colonize. Even with other tissues, there are studies ongoing in trying to introduce DNA into enough cells in the key organ that they would rectify genetic abnormalities.
So another question, a question from Alan Burgess, which I know you can answer because it's about, will you be sequencing human archaeological DNA? And I know that this is a really fascinating area because there was a conference I went to at the Sanger back in the before times in November, which was looking exactly at this, what DNA evidence has shed on human origins and evolution. So what kinds of samples now, and how far back can we go in terms of looking at ancient human remains? What can we learn from those, from the sequencing tools we now have?
Yeah, I mean, there's been tremendous advances in this. Almost, I don't think any of us could have really imagined it. So for, yes, sequencing Neanderthal DNA, sequencing, and then finding evidence of other ancient humans, the Denisovans, that we had no real idea of, that there's been no, I think, actual physical remain of. I think as I understand it, it's really about finding the materials from the right organs of the body. There are certain bones from which one can extract DNA, which would seem to preserve it compared to others. And there are certain environments that preserve DNA pretty well over time, very dry environments out in the dark that allow one to, so if one can find ancient DNA from those sorts of sources, one has the possibility to really go back in time and provide, instead of reconstructing what the DNA sequences of our ancestors were, actually getting bits of them in real generated sequencing.
Yeah, so if you can find it, you can sequence it.
More or less. The wetter I think it is, the less good it's just going to degrade. So you need to find it in those particular environments.
So from our ancestors to today, a question from Jonathan Chan, who says, given the pace at which DNA sequencing technology is developing, how long do you think it will take until everyone in the UK has their genome sequenced? And I will add an adjunct to that, which is, and is it a good idea?
You know, it's clearly we're on a trajectory to that. We've seen from the original reference genome, we went to the 1,000 genomes in 2008. The UK government announced its 100,000 genomes project in 2014. 500,000 human genomes are being done in the UK Biobank now, half of them here at Sanger. So we kind of, we getting towards having the technology that allows us to consider this. There are, you know, you can imagine a world in which actually it would be a useful thing to do to have that genome sequence, put it in your locker and have it under lock and key, perhaps your key, and you would let others look at it when it was going to be useful to yourself. When perhaps you might want to need an anesthetic and you would want to know whether you had some polymorphism, some variant in your genome that would make you sensitive, allergic to an anesthetic or to an antibiotic. Those are the sorts of situations in which having your genome stashed and ready would be a good thing to do rather than having to get it done on the fly. You know, we've been talking about this for a long time. The pace hasn't been, I think, as rapid as one might have thought, and the push to it. And I think much of that is because it's not completely clear that having your genome there would give you such great benefits. There would be some benefits to some people. We could all, or women could look to see whether they would have a predisposition to breast cancer, the rest of us to other cancers. But it's not completely clear that actually this is something that we as a society are ready to do now, or whether the investment in it now would be justified by the yields.
This sort of related a question from Sujath Abbas, who says, do we have any plans about making sequencing affordable in the context of diagnostics? So I guess it's kind of a similar question there, where there are a lot of moves in the NHS towards the idea of genomic medicine. Because we had the $1,000 genome and now we're coming down, you know, and how much do you think it will cost at some point for a genome, perhaps in a research context or a diagnostic context? You can put a figure on it.
Well, I think it's really is a question of how far in the future we look. I mean, at the moment, you know, the real cost of sequencing a reasonable coverage human genome is, you know, several $100s, a few $100s. We have learned that technology will continue to drive these things down. So it is quite likely that we will be confronting whole genome sequences of, you know, tens of dollars within the next decade or so. So those are absolutely kind of affordable for almost anybody. So it will be not a question, I think, so much of cost. It'll be a question of utility. It'll be a question of whether we can engineer systems that allow people to have their genomes safe, and to license usage of them when they need it or want it to be used. But one can absolutely see those things coming as we begin to add usage after usage after usage of the genome over the years. That sort of pattern, that accretion of uses will mean that it becomes more and more useful to have that genome just sitting there waiting for you to use it.
There's kind of two ways of going through this I've seen. There's some people who say, well, have your genome done and then you have it and you own it and you can then, you know, as we build our knowledge, it will become more useful. And the other idea that if it becomes so cheap to sequence your genome, then just have it done when you need it, and then like delete the data. And like when you need it done again, have it done again, because it's so cheap. So, and that kind of gets around a lot of the privacy and storage issues, I suppose. But
yes, that's perfectly
ways of doing it.
That's perfectly reasonable, and exactly, there will be many options. It's going to be cheap enough for us to consider all those things.
And of course, someone's suggesting putting it in a blockchain, but you know.
Yes, right.
But yeah, so a great question from Timwe. And this is talking a bit, I guess, about the themes that we picked up on sequencing the tree of life and whether this is in fact possible. Asks, how many different individuals of a species would you need to sequence in order to repopulate a species after extinction? So I guess the question is, if you had the sequence of an extinct animal, plant species of some sort, could you bring it back to life? I guess this is similar to what George Church is up to with his mammoth project. Do you want to talk about that, or shall I briefly?
I think you probably better talk about it.
So George Church, the US geneticist, is basically, they have sequences from mammoth cells that were frozen in the permafrost, and they sequence them. So they're trying to work out, okay, here's the mammoth genome. Can we engineer the important mammothy bits of that into the elephant genome and kind of recreate mammoths? Well, they're sort of, I guess they're just big woolly elephants at this point. They're not true mammoths. But is that something that would be feasible? If you have a genome sequence from something that's gone, can you bring it back?
You know, it's science fiction at the moment. Will we ever do it? I don't know.
I want to see a mammoth. That's
yeah, I don't know, really. I think we are a long way away from having the technology. That's an easy thing to say. It's an interesting idea. Probably not necessarily our best way of preserving biodiversity.
Oh, here's an interesting question. We've talked quite a bit about technology. So Lewis Strachan says, how important has the Illumina sequencing technology been in studying genomes? So maybe give us a quick overview of the difference between, we talked about Fred Sanger's original sequencing method, which was developed in the 1970s, and then this next generation technology and the sort of what Illumina managed to do. Can you just explain, like, what was that leap and how important this has been to make that leap?
Yeah. So the original sequencing technology was the one that was invented by Fred Sanger. It was very effective. It's what was used to sequence the reference human genome. Essentially, what one wants to do is to do that much faster and much more cheaply. And the inventions that went into the Illumina technology basically allow you to do it in a very multi-parallel fashion. So instead of having, you know, one sequencing lane, and I have a picture behind me where I've got sort of 96 sequencing lanes. Instead of having that, you have hundreds of thousands of sequencing lanes, but they are little colonies on a glass slide, and from each of those colonies you can build up a sequence and you read it from each of those. So it's like going, as I say, from having a sequencing production where you've got 100 lanes to having hundreds of thousands and just generating sequence from each of those. And that required some pretty fancy footwork in terms of technology and some really clever basic thinking that actually was invented originally in this country, in Cambridge. So that's what transformed it, and it's all about amounts of sequence and the cost of it, and that's what the Illumina technology allowed one to get. The changes recently in the Pacific Biosciences and in the Oxford Nanopore technology are these much longer reads of DNA sequence in the thousands and tens of thousands of letters of code. And that's the sort of thing that will make the big difference, well, in a number of different contexts, but in particular for sequencing the tree of life, that's going to be a critical element.
It is incredible. The sort of the analogy I like to think of, it's almost like the Sanger sequencing was like typing something out. You saw something, you had to type it on a typewriter, like one letter at a time. and then the next generation was just like photocopy the whole page.
Yes.
You just boom, 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 boom. It's that kind of scale. So we've, where are we going? So I think we're coming a bit to the end of the question. So I think this is a really nice one to finish. This is from Rebecca Hill. And, you know, we've talked a bit about what's in the science fiction realm, you know, recapitulating the mammoth. But what do you think will be the most significant advancements in this area in the next 50 years or so? So, you know, setting your realistic science fiction sights, what do you think is going to come? Or what would you really like to see coming?
Actually, I'm not, you know, I would sort of say that that I would prefer not to think about science fiction. And I would like to think about some of the things that are really coming along in the next 10 years, where I think genomes and genome sequences are going to be really quite influential. You know, we talked about human variation right at the beginning. You know, there are initiatives around the world which are really cataloging all the, well, as many as possible of the genes and the mutations within them that cause childhood developmental disorders, learning disabilities, autism, but physical developmental disorders. I mean, this is an initiative that is really been underway for many years. It's been contributed to here by Genomics England, by groups elsewhere, and this is the sort of collective endeavor of genetics using genome sequences that is probably going to give us a pretty comprehensive list of the genes which, when they're mutated in the germ line, cause some form of developmental disorder. This is going to be, and already has been, I mean, it's coming out and it's being used as it comes out. It's already of use to families who have children with these diseases because it allows them to ask the question, is this a new mutation or is it something that I as a parent have passed on to my child? Am I likely to have another child with this? And these answers are coming more and more clearly to parents, enabling them to make these sorts of decisions. This is very practical use of genomes and genetics, but is having considerable influence. That sort of thing will take another step, which we as a society will almost certainly have to consider in the next decade, which is that we already do test antenatally for certain genetic abnormalities, Down syndrome, for example. We screen for that. And women who have a child with Down syndrome can have a termination. As a society, we have licensed that. Well, with that full catalogue of genes which, when mutated, cause developmental disorders in children, it is quite likely over the next decade that we will be able to test for many of those. And the reason for that is because all our blood contains naked DNA. Our cells leak DNA as they die. And a pregnant woman's blood contains fetal DNA. And it is possible to sequence the fetal DNA from the mother's blood. So this is the basis potentially of much more widespread screening. Absolutely will need to be thought about with great care with respect to the ethics and, you know, in multiple different dimensions. But this, if it comes, is a very transformative change in the way that we manage women and their choices. And along the same lines, is really cancer. So this is in the same way that a fetus leaks DNA into the mother's circulation, cancers leak DNA into the person who's got the cancer's circulation. So it's naked DNA. And it has been a growing idea with, you know, growing momentum over the last few years, that actually could we detect that DNA from the cancer before the cancer has manifested itself symptomatically. And there are a number of initiatives around the world that are looking at that with some considerable depth and activity and energy. And it would not be surprising if over the next few years, just taking a blood sample, sequencing it for the naked DNA in the blood, trying to pick up the abnormalities that are present within cancer genomes, will become a way of detecting cancers early. If that does happen, that is a transformation in the way that we will be managing cancer. So these are actually very major changes that may be upon us from the usage of genomes, which are not in the realm of science fiction. They're in the realm of science reality, but really very dramatic in their impact.
I think it sort of highlights that the key defining feature of the human genome is that it's humans, and as a human society, we have to figure out, we've got these technologies, we're developing these technologies, who benefits from them, and what do we want to do with them to benefit humanity. So I'm very excited to see the next decade, the next 20 years of genome sequencing unfold. So thank you very much, Mike. This has been really interesting for me. I hope it's been very interesting for everyone listening at home. Thanks very much to the team at the Wellcome Genome Campus, to Beth and to Ken for organising it. And I hope everyone has found that inspiring night. Thank you very much, Mike. It's been a pleasure.
Thank you very much for inviting me. It's been great.
Thank you. Back to you, Ken.
Thank you, Kat. Well, I hope everyone found that as fascinating as I did. I'd like to thank Mike and Kat for such an interesting discussion on the science and its applications, and those ethical and societal issues that we all need to think about and take into account for the future of the field. It's been really great to hear such a diverse range of applications that genomics might lead to. As is always the case, there were so many questions, more than we could get through in the time, but hopefully we managed to cover lots of the themes that were coming through. If you'd like to be in the audience for our next event, please remember this is the first of 5 Genome Lates, the next being on August the 27th. We will have a panel event called Genomics Futures, which will feature people from across the Wellcome Genome campus, different skills, career stages and backgrounds, and they'll share why they got involved in genomics and their thoughts on where the science might lead to, and why the voices of everyone is important for the future of the research and its applications. So we'll put, Beth's going to put a link in the chat with the website to our events page, which is the Wellcome Genome Campus Public Engagement events page. At the bottom of that, you can also sign up to our events newsletter to keep informed about future events as they come up. We'll also put a link to the Sanger website. There was a couple of questions that wanted some more detail about the research and the publications. The Sanger was founded on the basis of open access and open data, and all the research you've heard about tonight you can explore for yourself. So we'll put the Sanger website up there and people can explore and find out more about the science using that. And also we've included a link to Kat's website where you can explore more about her current projects and our books. If you'd like to watch this talk again or share it with your friends or people who perhaps couldn't make it tonight, then we'll be posting it on our website in a few days' time, so you can look out for that too. Finally, as we close, we will post a short survey. Please, if you can, stay online just for a few minutes to fill that out. It really does. We value your feedback and it helps us improve these events and future ones. So please do that if you can. And finally, thank you again, Mike and Kat, and all of you for joining us tonight at our first Genome Late. Take care, and we hope to see you again soon. Bye-bye.